The Transmitter.
Here is a fresh episode of Synaptic. This is our show that looks at the people, the research, and the challenges of the neuroscience space. These are the researchers of neuroscience, and this is the work that they do. Episode 16 today. Thanks for joining me. Now, today, let's go back to 1996, and let's go to France. That year, France spent 2.22% of its gross domestic product on research and development. The United States that year spent 2.45% of its GDP on R&D. So that is not a tremendous difference, and of course, France is a smaller country than the United States with fewer scientists. But considering that the 1996 GDP of France was $1.6 trillion, US dollars, according to the World Bank, and for the United States it was $8.07 trillion, one can understand the difference in the total amount of money that was floating around for research back then in those two countries. So that's one metric of the times. But also, the notable research institutes of France were struggling. For instance, in 1996, INSERM said it was freezing 25% of its operating budget for its labs. And CNRS, the Centre National de la Recherche Scientifique, how was that, had been in a critical financial situation for the past 2 years, according to a news article published in the journal Nature. That was the environment in France when Catherine Dulac was finishing her postdoc at Columbia. That's our guest for today, Catherine Dulac. She had worked under Richard Axel at Columbia, and she'd published notable work while there. And she'd seen other people leave his lab and go off and start their careers, and she wanted to do the same. And she wanted to do it in France, in her home country. She had offers about everywhere in the U.S., she says. Harvard, for example, and MIT, and UCSD, and UCSF. But when she went to seek a position in France, she was told she was too young. Or, she said, she was offered maybe a corner of a bench in some lab. In essence, there were no independent positions for her in France, as she says on this podcast. So Catherine took Harvard's offer, and she stayed in the U.S., and she has been there ever since. We talked about that on this podcast. We talked about her discoveries in mice and the Vomeronasal organ around parenting and what suggestions that might have for our understanding of gender. And we also talked about her love of world literature as a child and how Marcel Proust's writing is related to neuroscience. All that coming up in the next hour and a quarter. I interviewed Catherine in her office on the Harvard campus. It was a nice summer day in Cambridge, high sun, but less than 80 degrees. Her office has wooden wall shelves filled with books and a sitting space centered around a kind of coffee table, and that's where I put the mics. But of course, just outside her office door, there were labs themselves, and the work was continuing out there, and so you can hear the low, constant hum of the equipment in the background. Okay, let's pick it up here where Catherine is talking about the new Dean of Sciences at Harvard, Jeff Lichtman, and how he is the first overall Dean of Sciences with a life sciences background. I think you're ready, so here's your episode of Synaptic with Catherine Dulac, starting right now.
Okay, so the history is the concept of having a Dean of Science is relatively recent, which is that Jeff is, I believe, the third or the fourth one. Depends how you calculate. But so there was a little bit of an intermediate period where there was a Dean of the Physical Science and a Dean of the Life Science. So here there was a Dean of Life Science. And then there were two Dean of the Sciences, and now Jeff is Dean of... So he's the third Dean of Science.
And the first one in the Life Sciences.
And before that, the faculty directly reported to the Dean of the Faculty of Art and Science. And then that became a little bit too much, I think. So now there is a Dean of Social Science, of Humanities, and of the Sciences.
Yeah. Okay, so you just mentioned you actually are not American by birth, though. You're French by birth, yeah?
I'm French by birth. I now have dual citizenship.
Yeah. Where were you born?
Montpellier. It's a town. It's an old town in the
That's
south of France.
Yeah, I was going to say.
Yeah, it's along the Mediterranean coast.
Okay, so how did you grow up? Were you interested in science as a young child? What was your childhood like?
Yeah, so I grew up, being interested in science, more generally just asking a lot of questions about how things work.
Yeah.
And then my two parents actually were scholars in humanity, so very different field, but they always encouraged me and my brother to read books on science, because they thought there was no job available for their students in humanities, and they thought,
they were professors.
In terms of finding a job, enjoying having a career, a scholarly career, if that's what we were interested in, then the sciences were a better bet. But overall, I think anything related to scholarship is of interest to me, or was of interest to me. We were living in this town close to an area where there are a lot of caves, for example. Caves, because it's a caustic geology, and so the water erodes and form these big caves, and many of them had been inhabited by prehistoric or ancient inhabitants. And for me, this was just fascinating. The idea of having these old ancient people living there. We were also looking for fossils always. There was a lot of old marine fossils or others. So for a very long time, I actually want to be a paleontologist.
Oh, you did?
But then I realized that meant, being with a little brush and then try to uncover little things. And I thought, maybe that's not that interesting.
These caves, so were these already... They were like historical. You could tour them. How did they know that they've been inhabited by prehistoric?
Well, there are a lot of caves of many different types. There are some that are just beautiful because they are all these formation, rock formation. Yeah. So I don't know if they were inhabited or not.
Oh, I see.
But they were just this absolutely humongous space that were just beautiful. And then there were others that, were on maps. And one I remember was, we had our headlamps. We were complete amateurs. We didn't do anything very difficult. But I remember I was kid, I don't know, maybe I was 8 or 10 maximum. And we came across some bones and some teeth. And so I took them, and they look like bones or teeth from bears. So I brought them to my teacher. Big teeth. Yeah. And the teacher was intrigued and looked, and they were from these prehistoric bears. I thought, oh my God.
Look what I've discovered, so right? Cool. This is so exciting. Yeah.
Yeah.
Your parents would take you and your brother out to do this.
Yeah.
Yeah. And so your parents were both in the humanities. Did they meet studying or something?
Oh, they met when they were students. They were students in Paris, and they met in whatever, when they were younger.
Students, yeah. So in Montpellier, there was a university that they taught at? So they were professors?
Mm-hmm. Yep.
Okay. So you did know kind of what an academic life was like. You knew your parents were professors. Okay. Mm-hmm. So you're growing up. You're interested in discovering things, it sounds like, the way things work.
Yeah, asking questions. I was reading a lot of books and always wondering how, why people were doing what they did. And this was more of, for the longest time, I just had this interest on how people think and what they do. I was an avid reader. I was reading everything. Sometime I would take a book and then I would go to my dad and say, I don't like that book. What is it? And he said, maybe you're too young to read it. And I would.
Are these biographies or historical books or fiction? Or
anything. My parents had books everywhere.
Yeah.
And so I would just go, we had bookshelves all around corridors, and I would just take books that seemed interesting. And, there was no age limit. I mean, that was my parents' books, and they had things of all sorts. And sometime I would, come across, philosophers or, really complicated things. And I'd say, I don't understand. But maybe you wait a little bit.
Yeah, you're not old enough. But were you reading like French authors? Were you reading fiction too?
My parents had books from literature mainly.
Oh yeah.
From all over the world. I mean, all in French, obviously, but they had section on French literature, on Russian literature, on Asian literature, on any type of literature. And in fact, I came to realize, coming to the US, for example, that very few people in the US have read foreign literature. For me, this was something completely natural.
Yeah.
I had to read the French, I had to read the Russian, I had to read the Japanese, I had to read the Africans, all of that.
So because it was all translated, you were getting fiction from all over the world.
Yeah. I mean, I read as a growing up, I read all the key, Russian, Japanese, what British, whatever, American.
Yeah.
For me, the fact that they were French or not French didn't really matter. Those were just absolutely fabulous books.
Do you think that had you, as you said, you're wondering like why people act the way they act or behavior? I mean, I could see how you could get that from reading world fiction.
Yeah, totally.
Huh.
Yeah, totally. And in fact, this is an aspect that I always found very interesting, is that why are you attracted to read novels? Well, it's about people, it's the life of people, it's the experience they have, it's their emotional reactions to events, and also encountering other people. And so for me it has always been very striking that a lot of our internal life has to do with how we interact with others. And one author that was extremely important for me is Proust.
Oh yeah.
Proust wrote these absolutely humongous books.
Yeah.
And I read them multiple times. It's like 7 huge volumes, and I read it, I don't know, maybe 3 or 4 times. And
as a child.
As a child, yeah. And for me, what was attractive is the introspection. Is the deep thinking of why am I reacting in particular ways. And somehow you can view this as the equivalent of what a neuroscientist asks. Why are animals, how is our brain reacting to environmental events?
Yeah.
But from that, humanity side of things.
So you're reading Proust, for instance. Are you asking the question, why are these characters doing the things they're doing? Or were you asking, why am I reacting to the story in this way?
All of it.
All of it.
Yeah, all of it. It's, it's that this internal life that we have, that others have, what are the guiding principles? Are there common principles? Or why are some individuals reacting to a particular event or a particular encounter in different ways? What are the rules?
Yeah. So I'm still struck by this. You read all of Proust like 3 or 4 times as a child.
Yeah.
That's amazing. Okay, so did you have any interest?
I mean, it depends what you call child, right? It goes up
to 18.
Yeah, absolutely. Yeah.
So in your childhood, you read Proust. I mean, most people, if they get through one book of Proust, they're proud of themselves.
I know. It's interesting because somehow what I encounter when I read a lot. There are books here, but that's nothing compared to what I have home. And I think a lot of people grow up and feel that they have to read things because their teacher is telling them what to read, right?
Yeah.
And then because it's an imposition, they maybe not realizing that there's so much joy in discovering books. And that's...
You mean because someone's saying you have to read this?
Yeah,
and then
exactly. And then they don't like to do it.
Yeah.
And then therefore they will never read anything else than what is being imposed to them.
Yeah.
So when I teach here at Harvard, I always put examples of books that I think illustrate in interesting ways some of the concepts. For example, I I have a lecture on schizophrenia, for example, and I cite a book on a family that has schizophrenia.
A fiction book.
Yeah.
Oh, you do.
There's I talk about smell, and I mention a book in which smell is important. Actually, Proust is one of them, but I don't cite that because I know that people are not going to read it. Or a book about vision, and I cite a book from Saramago on blindness and seeing. And I hope maybe even if one student take the book and read it, out of my entire class of 100 students, that's
a victory.
Win. Yeah.
That's a win. Yeah. One more thing about Proust. I mean, Proust has that famous scene where the boy's dipping his cookie in the tea, and it opens up all of his memory. That's a very neuroscience.
Totally. In fact, it's repeated over and over and over again in the field of olfaction, as how evocative smell is of ancient memory. And in fact, it's interesting because there are other examples of that type in Proust. And for me, actually, the most striking one was not that particular example of dipping his Madeleine in his tea. There is an example of him walking around, I think, the house of his grandmother, and there are these bushes with flowers, and the smell of these flowers is also extremely evocative. So when I started to work in the field of olfaction when I was a postdoc, and at the beginning here in my lab, people kept bringing the example of the Madeleine, and it's interesting because that's the example that is the public example, but actually there are many other ones which I think are even better, but nobody has read them. So
they don't know. Yeah. So one more thing. Did you, with all this reading, did you harbor any interest in being a writer before you went off to school?
No, never. I'm a consumer.
You don't want to produce it. You want to consume it.
It's interesting. I never thought about it. I love poetry also. I don't think I've ever even tried to write a poem.
Yeah.
I don't think I would be very good at it.
Yeah.
But
okay.
I just enjoy it.
Yeah. Okay. So I guess the question is, when you go off to college, and that's in Paris, right? That's the École Normale Supérieure.
Yeah. Mm-hmm.
Did you were already thinking about being a scientist or what?
Yeah. So my interest in biology started in high school. Actually, high school or mid school, I don't recall. I had a teacher, like many people, your attention, you get attracted to a field because you just have a fabulous teacher.
Yeah.
And there was this teacher in biology who, was just describing something, I think about how the cells are organized, something like that, some really basic biology. And I asked, why is it that way? And he said, in biology we never ask why, we ask how. And I thought, this is so interesting. And I don't know, somehow I remember that time as sort of almost switching something in me and emphasizing that, there's so little we understand in biology. We still have a lot of questions to answer of the how that works.
Yeah.
And I think I got attracted to biology because of that.
Yeah.
From that time.
Yeah, that was feeding your natural curiosity that you had already up to that point. Yeah.
And in fact, some of the knowledge that I acquired in becoming a biologist was not only strictly biology, it was also geology and anything that is related to the world around us. I just loved it.
Yeah. Okay. So then when you finish your undergraduate degree, you rolled that right into a PhD, I think, right? University of Paris?
Yeah.
And there you did study developmental biology.
Exactly. Yeah. So as part of the École Normale Supérieure, I should actually back up a little bit. One thing that really interested me was the idea of doing research, of trying to explore something, discover something, still this question of how instead of why.
Yeah.
And everywhere I went as a child, even my parents told me, it's very difficult to be a professional researcher. There are very few positions of that type, so don't get your expectation too high. You might succeed, but chances are it's not going to be possible. And I think I just enjoyed so much learning that I didn't really think about the career option in a very serious way. I just thought, I'm
enjoying myself. Yeah.
And so that's what I did. I learned biology. I got lucky to enter into this very elite place of École Normale Supérieure, which is totally geared towards research. And that gave me an entry into a lab as an undergrad. So I did already some research as an undergrad, and then I stayed in that lab to do my PhD.
Oh, you did? Okay.
Yeah.
So that was with Nicole Dorin, right?
Yep. Mm-hmm.
Were you seeking her out? Did you know her work or?
No, I think that she contacted someone at the École Normale and say, I'm looking for a graduate student potentially or an undergrad that would be interested, and so they are opening in my lab basically. And so then I went, and I thought it was super
well, that
interesting.
person suggested you?
Yeah.
They knew something about you that thought you might fit with Nicole.
Yeah. Well, one thing is that she was one of the very few head of laboratory women.
Oh, yeah.
So, she was interested in having women students.
Yeah,
and
yeah,
so
smart.
That's,
Okay. So when you finished your PhD, then you had some decisions to make.
Well, you're going through the PhD a little bit too quickly, because the PhD, first of all, was for me a revelation, because I was learning the job of being a scientist, and I just loved it. But the French system is a little bit different here. Because my PhD was successful, I got a position right away.
Oh, okay.
A lifetime position. So that's it. I could stay in that lab forever.
Oh, I didn't know that. I didn't know that.
And so, but I didn't feel I should do that, because I thought I had to go somewhere to learn something different, to just expand my knowledge. The other thing is, I had never learned English in school. I learned all sorts of language, Greek and ancient Greek and Latin and German and all of that, but not English. My parents said, you're going to have to learn English anyway, so why don't you learn other things first? And I thought, maybe I need to go to the US or to the UK and, being in an English-speaking country. And this plus, the American scientific system works so well, so efficient, so productive. I thought I had to come and experience this directly.
Okay, so you had an opportunity just to start a lab. No, not in the US.
French system, you don't start a lab. You're in somebody else's lab, but you have a lifetime position.
Oh, I see what you mean. Okay, so yeah. No wonder.
Well, you said no wonder, because you are you're used to the American system.
Yeah.
But in the French system, it doesn't work that way. Scientists are part of the these are government positions, and starting your own lab is actually not easy.
Right, but I'm saying, did you want to start your own lab, or you didn't even?
Oh, so that's an interesting question, because in retrospect, I was just so oblivious. I never thought about starting my own lab. I thought this was completely out of my reach. As with my studies, I was just enjoying myself doing research. And when I was, as a PhD student, I was looking at the PIs around me and said, oh my God, this seems to be so out of reach, so difficult, and you need to find funding, and you need to find projects for people and all of that. Oh no. And so then I went for a postdoc, and I also had the same, I don't want to say fear, but I really thought that it seemed so incredibly difficult to be a PI. And I didn't think I would necessarily be able to do it. And it's only completely at the end of my postdoc when I made a big discovery, discovered the genes encoding pheromone receptors, and that suddenly opened all sort of new question, new horizons, and at that time I thought I could do it.
We should say your postdoc was at Columbia.
Yep.
Yeah. So you had not studied English at all, and you came over to the U.S. to start that postdoc?
I had studied English. I had a book and I had some tapes, and every day for half an hour I was learning a lesson when I was a PhD student. And then my PI, Nicolas Doran, sent me for a collaboration at Cold Spring Harbor. So I spent a summer there. But I remember, I was like a fourth year graduate student or something. I remember landing in New York, coming down the stairs of the plane and thinking in my head, I have never spoken English to anyone. Like, in my head, on the tape, but never to a real person.
Yeah.
And it was actually quite interesting because for some reason, I think I chose the right textbook or whatever, or because it was American English rather than British English.
Yeah.
And I was with a lab mate who had learned British English in school. And actually, I could understand way better than he did.
Oh, really?
Yeah. And I could say things that were, more, I guess, American. Than I remember actually staying. I think I stayed 2 months or a month and a half, so quite a long period of time. And at some point, I realized that I was dreaming in English.
Oh.
And that was super surprising to me.
Yeah.
I thought, oh my God, I must be dreaming in terrible English. But who cares,
Yeah,
right?
that's a dream. What does it matter? So with your postdoc, you're with Richard Axel.
Yeah.
Right? And so he later, right, he would go on to share the Nobel for odorant receptor genes, right? So he's working in olfaction as well.
Mm-hmm.
Did you know that going in?
Yeah.
Okay, so that's what you wanted.
Yeah, so I did my PhD in developmental biology in an absolutely beautiful system, experimental system, which is the quail and the chicken embryos the cells are able to mix, and that enable to recognize the migration of cell types within an embryo. So it was, intellectually and experimentally a very beautiful system. But somehow I realized when I was a graduate student that I was very mechanistic in sort of more molecular ways. And, there's no genetics in chicken and quail embryos. There's not a lot of molecular biology. In my PhD lab, I was the first person to ever purify a protein, the first person to ever make a cDNA library, the first person to clone a gene. And I just want to continue along those lines. And so in 1991, the paper from Buck and Axel came out, the cloning of olfactory receptor gene. And I thought, this is fabulous, because here you recognize the identity of a particular olfactory neuron type based on the expression of its olfactory receptor.
Yep.
Every type of olfactory neuron expresses one particular type of olfactory receptor. I thought, this is fabulous. It's like a new sensory modality is now open up to molecular biology and to developmental studies using molecular tools. So that's why I went to the Axel lab, because I was attracted to that. And I had two projects. I had a project that was my main project that I thought was going to be very difficult, maybe impossible, and then a side project that I thought would be simple. And both of them ended up very difficult. The first one was to understand the development of olfactory neurons. And the reason I knew this was going to be difficult is because the way people had understood how T cells and B cells in the immune system were able to choose a T cell receptor or an immunoglobulin is by making cell lines out of B cells or T cells. And so you take these cells, you make them immortal, and then you see whether these particular molecular mechanisms that enable a B cell to express one type of immunoglobulin or a particular T cell to express a particular T cell receptor. And because you have a clonal expansion out of a single precursor, you have a lot of material, and you can see whether, for example, the genome has rearranged, which is the case of T cells and B cells. But because I was trained as a developmental biologist in the nervous system, I knew that it was almost impossible to make a cell line out of a neuronal precursor. Either a neuronal precursor divide, but then it's not differentiated, it's very unlikely to express olfactory receptors, or it's differentiated, but then it cannot divide anymore. So the idea of to have a clonal expansion of an olfactory precursor seemed impossible. But I was going to try. And this was way before the iPS cells or, neural stem cells, etc. So that was the difficult project. And then there was the easy project that ended up being not easy. That was the cloning of the pheromone receptors.
Right, okay.
And the reason everyone assumed it was easy is... you have to imagine, this is just few years after the discovery of the olfactory receptors, which was, magnificent discovery. And so there was absolutely no doubt that the pheromone receptors would just be, a subclass of the olfactory receptors. Therefore, all I had to do was design primers or molecular probes that worked on olfactory receptors and find something that was a little bit on the side. It ended up being not this at all. But at the beginning, that was my easy project.
But you said when you first got there, too, you were looking around the PIs, maybe at Richard, and thinking, I don't know that I can do this. Did that feeling continue?
Oh, yeah. Continued up to basically the year before I went on the job market. Because it seems so difficult. I mean, both my two PIs, Nicole and Richard, were people who were incredibly smart, incredibly successul, knew a lot of very important people, knew what to do, basically. And I think that as a graduate student, as a postdoc, I thought, oh my God, how do I know all they know, what to do, all the people they know?
How do they meet all these people? Plus running the lab, right? I mean,
running a lab and have an intuition on what project to pursue, what project to stop. How to apply for grant, get funding for everyone. It seems very overwhelming. It turns out that seeing people who were more senior than me make that transition and being successful was actually very helpful, because if they can do it, maybe I can do it.
Oh, you would see someone else in the lab leave and started their own lab.
And because that's the American system, which is postdoc.com, and they stay for a number of years, and as soon as they have a paper or two, then they apply for jobs and they are hired. And so seeing this, that it's actually a normal cycle, made me more confident that I could do it.
Yeah, it would happen to you too. Yeah. But you said both these projects ended up being really difficult. So for a while, were you thinking, I'm not gonna make this, I'm not gonna be able to, or no?
Oh yeah. It was very stressful. Nothing was working. Nothing was working for a number of years.
Oh, really?
And, but that, to some extent, that's also part of research. You try something, it doesn't work.
100%.
So you try again, it still doesn't work.
Did you know that, though? Were you like, did you know that it's okay to fail for a couple of years and not get it to work? Or were you worried about that?
Oh, I was extremely worried, obviously. I think that Richard had a very nice way of reassuring people. Because he was saying, people in my lab try to tackle extremely difficult problems. And he had confidence in himself, in his ability to pick people who could solve those difficult problems. And so he say, I'm very confident you're going to be able to crack this open. I don't know when, but I think you can do it. And so it was interesting to hear, because if you think about his history, first of all, he has this incredible lineage of people who were trained in his lab. David Julius is one of them, but Linda Berg is another one. But, he has this just amazing former trainees that each would come in his lab and publish on a different topic and then take this over. And olfaction was the first topic he actually kept in his lab. In the past, people would just leave and he would switch to something else. And so this also was part of helping, is to think that he has confidence in me being able to solve that problem. This being said, what happened is that I was trying to clone the pheromone receptors, again, based on similarity with olfactory receptors. Everything I tried failed. So I would say, okay, well, maybe I didn't do that well, so let me try in a different way, and then in a different way, and then in a different way. And then at the end, I thought, okay, maybe I might be incompetent, but not that incompetent. And I think there's something just fundamental that I'm not understanding. And the fundamental assumption I was making is that these pheromone receptors were similar to the olfactory receptors. And I thought, maybe this is just wrong. And it's actually easy to test, because in the olfactory system, the signaling pathway that is triggered when an olfactory neuron detects an odorant, this was known. It was known that the exposure of olfactory neurons to odorants in frogs or other animals that were tested lead to an increase in cyclic nucleotides. And so therefore, the idea is that there is a cyclic nucleotide gated pathway, meaning the receptor has to be a GPCR. So GPCR, 7 transmembrane domain receptors being activated lead to the activation of adenylate cyclase, lead to the increase of cyclic nucleotides that open a channel, and then you excite the neuron, you send the information to the brain. That was known for the olfactory system, and so that was the basis of the discovery of the olfactory receptors, because a lot of 7 transmembrane domain receptors were known. And what Linda Berg did in an extremely smart and ingenious way is to design primers that would recognize any type of 7 transmembrane domain receptors. And all she asked, she had a number of criteria. One is that whatever she found had to have 7 hydrophobic stretches. They have to be specifically expressed in the olfactory system, obviously. And then they had to represent a large family, because the olfactory receptor had to be very diverse. And she found those receptors. But now, when I looked for the olfactory specific cyclic adenylate cyclase, the olfactory cyclic nucleotide gated channel, all of that, all this olfactory signaling pathway was not expressed by neurons detecting pheromones. In other words, I came to realize that these receptors could be of any type. They could be tyrosine receptors, they could be GPCR of a very different sort, they could be anything. And so all the strategy that I had used so far were based on having some intuition about the nature of these receptors. And now that was gone.
Is that because the nature of a pheromone is so different in the body? Like what a pheromone is meant to do in the body is different from a normal odor.
Yeah, so odorants it's the sense of smell, right? It's this information about volatile chemicals that provide information about food, about danger, about smoke, about all of that. Pheromones are semiochemical, so they are signals that are exchanged between animals to provide information that then lead to specific behaviors, social behavior in particular, which is what I was interested in. In mammals, semiochemicals are typically not volatile, which is, if you have a dog, or if you see what dogs do, they put their nose in the butt of another dog, it's because they touch pheromones, or the way they smell the sidewalk, they put their nose. So you need a contact, physical contact, because pheromones are water soluble. They are in urine, they are in sweat, they are on the skin, so there's a contact that is needed. So you can imagine that the receptors for those molecules are completely independent, have really evolutionary no similarity whatsoever with odorant receptors.
Huh.
Also, the pathway in the brains are completely different. So the main olfactory system, the one detecting smell, goes to the olfactory bulb and then goes to 5 or more areas that overall comprise the olfactory cortex. And that leads to the conscious perception of a smell. What you smell of flower, you smell coffee, you smell wine, you have a conscious perception of it. But the vomeronasal organ, this set of neurons that detect pheromones, project to a particular area of the olfactory bulb, and from there completely bypass cognitive areas. And so they go to the limbic system, to the amygdala, to the hypothalamus. And the idea is that they trigger instinctive behaviors. And for me, this was the attraction to the pheromone world.
Right.
Is
so this
to
is
have access.
We're beginning to touch on your work that you're doing at Harvard back in the early 2000s, right?
Mm-hmm.
Yeah. Okay. All right. So then, when you finally do have your success here, at Columbia, and you had a big paper out of that too, I think. Yeah.
Mm-hmm.
Then it was your time to move on. Did you think about going back to France, or did you?
I wanted to go back to France. In fact, when I went to the US, there was absolutely no doubt I was going to go back to Paris within a few years. I had absolutely no intention to stay in the US. I'm French. Why would I stay in a foreign country?
Yeah.
But then, first of all, the American system has something very special, I think. It's successful for very good reasons.
Yeah.
It's the way it's organized. And it was a difficult decision. I tried to find a job back in France, and there was no independent position for me. And it was interesting because I had offers about everywhere here that I applied to. MIT and Harvard and UCSD and UCSF. All those places I had offer. And then I would go to France and would say, "Oh, you're way too young to be independent," or you cannot have your own independent budget, or here's a little corner of a bench that you can have. And the discrepancy was like, what the heck? There's no way I can go there. And so the American system is extremely attractive for me as a way of being organized, because places like Columbia or Harvard or UCSF are these large academic institutions where there's a large number of scientists. There's this really interesting collegiality and collaboration.
Yeah.
And so this is where I would say the academic world that I knew in France was quite different than here. The scientific community in the US is very large, and so it's very diverse. Well, maybe not that diverse in certain ways, but anyway, there's a lot of different type of science occurring, and it's very enriching to be part of this community. In France, maybe not only in France, in other countries in Europe, the communities are much smaller. The availability of funding is also very reduced.
Yeah.
And so as a result, there's way less communal enterprise or collaboration compared to here. So this is a very attractive academic environment for me.
So it's you made this bet that you sacrificed your own culture for science, science life. Yeah. Okay.
Well, not really, in the sense that everything went by step, which is that I came to realize that if I want to start a lab and have my own project, I could not go back to France. Going back to France meant basically completely changing my plan. When you do a postdoc in the US and you're successful, it means you have an entire new field that is opening up thanks to your discovery. And so, you're on a roll and you want to do experiment along those lines. And it was clear that was not possible in France. There was I needed to have transgenic animals, mice, large mouse facility, all of that. That just was not available to me. So I would have to completely change. To do what? Well, to probably work on the project of somebody else.
Yeah.
And that was not attractive. And so I thought, in the meantime, I had great offers in many places, including here. I thought maybe what I'll do is I start my lab as a junior faculty. I learn how to be a scientist as an independent junior faculty, and then I go back to France. And what happened is that my lab worked really well. We had a lot of great publication almost immediately. And so I got tenure very quickly.
That's like 4 or 5 years.
Yeah. And then, okay, I kind of got used to the good things. It was just not possible for me to move out because, things were going so well here.
Yeah. So where's your brother, by the way? Is he in France?
He's in France, yeah. He's
and
a
your parents
physician.
were in France?
Yeah.
Yeah. So,
yeah.
That's a big, I'm sure you got to see them, but it meant a long flight, right?
Totally.
Yeah.
So,
okay.
being exiled to some extent, it's not always very easy.
Yeah. So you joined Harvard in 96 or something like that?
Yeah, fall 96, yeah.
And did you have postdocs, or did you have grad students immediately?
Yeah.
Oh, so how big was your lab?
Well, so there was 2 postdocs who joined right away. And then the first year I actually had 3 graduate students joining, and then 2, and then so my lab became 6 to 10 in the first 4 or 5 years.
Wow. Okay. So it's because you were, as you said, having these great publications. That's how you got tenure so quickly?
Yeah.
You just were producing.
Yeah.
Wow. Okay.
And people got scared that I would leave. So,
oh, really?
Yeah, I think American institutions are terrific for that. I mean, Harvard really helped me. They want to keep me. They helped me buy a house, and they, gave me this great lab and funding. And why would I leave?
Were you being wooed? Were other schools coming at you saying, maybe do you want to set up a lab here? Harvard, if they were nervous.
Yeah, but not in a formal way, because the reality is I was doing well here. I had no desire to go somewhere else.
But they must have got wind of that somehow. If they were nervous you were going to leave.
Probably.
Yeah. Well, must be glad you stayed.
Yeah, I'm glad. I'm very happy here.
Well, let's talk about the vomeronasal organ, right?
Okay.
So that really took off in your own lab here at Harvard, yeah?
Yep.
So tell me about that work.
Well, so I came to realize that a lot of assumptions that we had about the vomeronasal organ were wrong. And so I think this was some of the just great enjoyment also of doing research, is to discover that all your expectations were wrong. And so then, nature is always more interesting than what you can predict. So to start with, in Richard Axel, I discovered this family of pheromone receptors, and then realized that that was just taking into account a particular subset of the neurons, and so we discovered a second family of pheromone receptors. So now we have these 2 large families, and I think that there are a total of 300 receptors or something like this. And these, to start with, was way more than what we had expected. Because the idea is that the vomeronasal organ triggers mating behavior, aggression, a number of stereotyped social behavior in rodents. And so maybe how many receptors do you need?
Yeah.
I don't know, maybe 10 would be enough, or 20. So now we have 300. So that was problematic to some extent, because initially the plan was, let's discover the mating receptor or the aggression receptor or the parenting receptor, and then let's do genetic manipulation of those receptors to know how they encode that particular information that lead to a specific behavior. With 300 receptors, this is impossible, right? And so...
Were you going to try to knock them out one by one in mice, or what were you going to do?
Yeah. For example,
so
to knock them out, to look also where they project, try to understand what they do, how they do it, right?
Yeah.
But with 300, this is impossible.
Yeah.
And so it meant that the system was way more complicated than what we had anticipated. And so that led me to collaborate with Emily Lyman, who at the time was a postdoc with David Corey here at the medical school. And she's a physiologist, and she had this really interesting idea on vomeronasal signaling. If it's not cyclic nucleotide based, then it has to be of a different type. And the only other type that was known was the one related to Drosophila vision, which use TRP channels, which is a different type of pathway. And so she had the idea that maybe, and that the idea is really coming from her, that maybe the vomeronasal signaling was based on TRP channel. And so, because she's a physiologist, I'm a molecular biologist, we decide to collaborate, and we indeed identify specific TRP channel, particular TRP channel called, at the time we called it TRP2, now it's called TRPC2, that was extremely highly expressed in the vomeronasal organ, and when we made an antibody, it was nicely expressed, the protein was nicely expressed at the sensory terminals of the vomeronasal neurons. So Emily went on in looking at the physiology of the neurons, and I used that gene to knock out, to impair genetically vomeronasal signaling. And that was, the huge surprise, is the VNO was actually, the vomeronasal system was not doing what we expected it would do. The expectation, again, was that this is the trigger for mating behavior and aggressive behavior. But when we look at mutant males for the vomeronasal system, vomeronasal organ, males were able to mate perfectly normally. In fact, it was interesting because Richard Axel, also in his lab, had also done that knockout and couldn't find any phenotype. And so the students was doing the work basically left the lab and went to medical school. And I was very lucky to have, as a postdoc, Lisa Stars, who is now at the Scripps in California at UCSD, in San Diego, who say, well, if the genetic impairment of the vomeronasal system does not affect mating, let's look at another behavior that we think is pheromone mediated. That's male-male aggression.
Yeah.
And so she put two males together. One is a wild type, one is a TRPC2 mutant. And, we want to see whether they would fight or not. And the mutant male tried to mate with the other one. So, oh my God, what's going on here? And we realized that the mutant male was basically unable to discriminate between a male and a female, which is, if you put a male and a female in a cage, the male mutant would try to mate with the female, and then the male, and the female, and then the male. So what the vomeronasal system does is not to trigger mating behavior, is to discriminate between males and females, and basically tell the animal either you mate, or you attack, or you do other things.
Yeah.
So it's more like a switchboard rather than a trigger. Simple trigger of and so that was super, super interesting. And then there was this really interesting period where I would describe this work, and then when I would give a seminar, there would always be somebody raising their hand and say, okay, so this is the phenotype of the males, but what about the females? And so here I came across a really interesting phenomenon that surprised me at first. Now I think that this is well recognized, is that actually people had strictly no interest in the behavior of females. There was very little in literature. So I was lucky, because here again I had a terrific postdoc, Tali Kimchi, who is now professor at the Weizmann, who say, I'm going to look at the behavior of females, and she was used to the behavior of wild animals, and so she thought she could discern if there were even subtle differences. So she looked at the mutant female, and she just wanted to do like a very simple test to see whether that mutant female was sexually receptive or not. So she put the mutant female together with the male.
With a wild type.
Again, the female mutant tried to mate with the male with a male typical mating behavior. It's like the female was behaving like a male. So, wait a minute, check the female. Yeah, it's a male. It's a female. It's not a male, but it was behaving like a male. And so we thought, maybe the circuitry is developed abnormally because the animal is a mutant, never had vomeronasal input.
Right.
So we did. We did an experiment that ended up being very difficult, which is to remove the VNO surgically of an adult, and we found exactly the same phenotype as the genetic mutant. But in the meantime, we also discovered something that explained a lot of the old literature that say that the VNO is essential for mating, which is that when you remove surgically the vomeronasal organ, so the VNO is in the septum here, in the cartilage in the middle of the nose. When you remove it, what happens is that there's a lot of bleeding. And what Tally discovered is that in the majority of the surgery that she did, the nasal cavity was basically full of blood, so was completely clogged by blood. And so she had to do all sort of trickery to clean up the nasal cavity after the surgery, to make sure that the VNO was removed, but the main olfactory system was accessible. But when people had done the surgery in classical work, they didn't check that. And so what happened is that these animals were both deficient in main olfactory system and vomeronasal system. It turns out that if you look at an olfactory mutant, they have great difficulty of mating. So the olfactory system, the main olfactory system, is actually essential for mating behavior. But people never realized that because they were so focused on odorant reception. The VNO does the instinctive social behavior. The main olfactory system does smell. Mm-hmm. And people had olfactory mutant lines that were extremely difficult to breed. But they never associate both. Oh, maybe the olfactory system is essential for breeding. They never did, because it was known, that was the assumption, that the olfactory system was essential for smell, not mating. So it's interesting how we get stuck in these preconceived ideas that end up being completely wrong.
Yeah.
So this idea that females now behave like males if they don't have vomeronasal input was very influential for my own thinking, because first it indicated that if a female is now able to switch to a male behavior just because we remove a piece of the nose, it means that the central circuitry is there, right? And so people hated that idea when I first published it, because the textbooks say young males have a surge in testosterone that makes the brain circuitry male, and that's it.
Right, so they develop, once the hormones come in, they develop differently from that point on.
Exactly. You have a male brain and a female brain, and that is not the case.
Yeah.
There's a brain, and that brain is suddenly influenced by hormones in very significant ways, but the hormones act on circuitry that do exist in males and females.
When this kind of work is done, where when you read it, you can't help but think of humans, right?
Yeah.
And do you think that's why people get resistant to ideas like that?
Well, people get resistant of ideas because first that contradicts a lot of work in animals. And the problem is there was a lot of reluctance to accept some work that were basically challenging ideas that had been in the field for over 50 years.
Yeah.
Those ideas were done in a particular experimental paradigm. I was invited at the time we made this discovery in 2007, 2010. I was invited to give a seminar in the endocrinology department here at the medical school at the Grand Round. So there was a lot of physician endocrinologists over there. And somebody came at the end of my talk and say, oh, I thought your talk was really interesting because that person was the director of the transgender clinic. And he said that he was just starting in that position, and he said that he was very surprised because he had assumed people who would go to his clinic would be, teenagers that have, these hugely fluctuating hormones and don't feel good in their body, etc. And he said, no, the patient I see are kids that are 4 or 5 year old. They barely speak, and they already say, I know I'm not a boy, or I know I'm not a girl. And so this has nothing to do with sex. It has to do with your gender identity, something that way precedes any type of sexual behavior. And so if you think that there's a male brain and a female brain, then this transgender phenomenon is a little difficult to understand. If you think that there's a brain, that then there are all sort of regulatory mechanisms that makes a male to behave mostly like a male and a female mainly like a female, then the phenomenon of transgenderism is a little bit easier to understand because you can understand these differences and whatever phenomenon occurs. And so it was interesting because I remember I still have the slides, and I found that really interesting. So I was invited to give the keynote lecture in the big endocrinology conference in 2008. And I presented those data. And I remember how much of a cold shoulder I got.
Oh, really?
Yeah, clearly. I mean, 2/3 of the people just didn't buy what I had to say.
You mean afterward?
During the conference, and then, when we published our paper, I got some letters, very condescending letters from endocrinologists who were telling me that they would be happy to explain to me how to do behavioral experiments, because clearly I didn't know how to do them properly. It was very offending. But then I got also invited 10 years later, 2018, I still have those slides, also to give the keynote lecture at also this big endo meeting. But this time there was 3 keynotes. There was me talking about sex-specific behavior, the control of sex-specific behavior. There was a physician who was treating transgender patient, and then there was a transgender person who was a physician and was talking as a transgender.
Both as a physician and lived experience.
Yeah. And so things had turned entirely. That now this concept that the circuitry in the male and the female brain are largely shared instead of being, distinct, was becoming attractive because it made so much sense.
But the other thing is, we don't have a VNO.
No.
So, but these things that you were seeing in mice, they don't actually translate to humans because we don't have that.
So this is a very important point, and thank you for bringing this up. So I think what is extremely important to realize is that there are different levels of information that give rise to specific behavior. One is the sensory information, right? The external information. And what is extremely important to realize is that every animal has its own idiosyncratic ways of understanding the world, right? If I'm a mouse, I rely on olfaction and on the Vomeronasal system. If I'm a bird, I might rely mainly on sound or vision. And so, a mouse is not a rat is not a human or not a primate. Every animal has its own idiosyncratic way to receive signals. And you want this because you want to be able to recognize signals that matters to you, the one from the one that don't, right?
I see what you're saying.
And so, for sure, the Vomeronasal system has nothing to do with human or primate in general, but neither with birds or neither with a lot of animals, right? So every animal detects their species in particular ways. It's interesting, if you look at the brain of a mouse or rat, about 2/3 of the brain is occupied by olfactory-related areas. If you look at the brain of a human or higher primate, about 2/3 of the brain is dedicated to visual processing. So the importance is not the specific sensory information that comes. The importance is the circuitry within the brain. And here, again, the circuitry of a mouse is not the circuitry of a primate or human. There's absolutely no doubt. But the template is the same. So whatever we learn in the mouse probably can at least inspire research or finding in human. And so the finding we had made with these females that mated like males when they were mutant, then raised the immediate question about, if the female has male circuitry, does male has female circuitry? And so that's how we turned to parenting behavior. Because parenting was considered to be the female, the mother, the female specific behavior. And we thought, if we can show that the Vomeronasal system is essential for female to be parenting and male not to be parenting, then our reasoning might be correct. And so in mice, virgin males are typically non-parenting. They basically attack and kill mice. Whereas virgin females, they are parenting. They are not extremely parenting. They are not like mom, but typically after few trials, they will retrieve pups and bring them to the nest.
Right, so pups that are not their own, they will
pups
nurture,
that are not their own.
But the males will go after it.
And kill them.
And so but that's like an evolutionary thing, right? We want to remove it from the gene pool. That's why
yeah, exactly. So Sarah Hardy, who was actually a PhD student here at Harvard in the late 60s, discovered this, not only discovered actually, because it was described already, but she gave an evolutionary explanation to the phenomenon of infanticide behavior. She was observing langurs in India, and she found that each time a male would enter a social group, foreign male, then all the infants would disappear or be killed or wounded. So she never actually observed an actual attack, but she inferred that the male were actually getting rid of all the infants, and then the female, now no longer having offspring, then would be sexually receptive again, and the male would be able to mate.
Right.
And so her theory was encountered with a huge uproar because she was basically saying infanticide behavior is a normal behavior. And people say, no, there's just no way. Why? Well, it's interesting because she was in the psychology department. So there are two types of people who study primates. There are people in zoology department who look at primates for primate, and the people in psychology department who look at primate as a proxy to look at the evolution of human behavior. So here is somebody who was saying, infanticide behavior is a normal behavior. No way. And so there was this huge uproar. This can't be real, male attack infant because they are loud or because they are hungry or, all of that. But then people actually confirmed her data, found infanticide behavior. I think the next species were lions. When there is a foreign male entering a pride of lion, first thing they do is to kill all the cubs. And then rodents. And so this became an accepted fact that infanticide behavior is a normal behavior in some species. About, maybe 40 to 50% of mammalian species are infanticidal.
Okay, so a couple things I want to ask you still. One is you won the Breakthrough Prize in 2021, right? And this is basically for the work we just discussed.
Yes, so the work I got the Breakthrough Prize was the discovery of neurons driving parenting behavior.
Yeah.
And so again, we haven't really discussed that yet, but basically once we knew that males could be parenting if you remove the vomeronasal system, so that's exactly what happened. The vomeronasal system was again enabling the animal to behave either as a male or a female. If you remove the vomeronasal system, then male can be parental. So there has to be neurons driving parenting behavior both in the male and the female brain. We look for them based on activity markers, and we found a population of neurons in the hypothalamus expressing galanin. And we did all sort of genetic manipulation to show that these neurons do indeed are both necessary and sufficient for parenting behavior both in males and in females. And that made us realize something that had been described in the literature in some obscure journal, is that indeed males do become parenting after they mate with a female. And so there's this extremely interesting switch in behavior where males mate with a female and then 3 weeks later are actually paternal.
It's this short period while they're preparing for their own pups.
Exactly.
Right? Yeah. And then that goes away after the pups reach a certain age.
Yeah. Mm-hmm.
Yeah. Okay. And so, but I was looking at that. That was 2021. So, I know a little bit about the Breakthrough Prize and the ceremony and stuff. Well,
actually, I got the Breakthrough Prize during the pandemic.
I was going to ask.
I never went to the ceremony.
They didn't have it.
I have that thing. Where is it? Oh, it's here.
Oh, that's it.
Yeah.
Wow.
They sent it to me.
So, part of the goal of that is to mix like science and celebrity and make them all. I know, but it's a shame that they didn't get to mix anybody.
I have to say, they gave me a chance, but.
What do you mean they gave you a chance?
Well, so during the pandemic, there was no ceremony, but then last year I could go and get my ceremony, but that was the 1-year anniversary of the passing of my mom, and so I went to France, stay with my dad instead.
Okay,
yeah.
Well, I think you made the right choice there.
Yes, I think so too, but that's okay.
You're doing often behavior work. It looks like behavior work, but you're coming at it from a biological standpoint, and I think that's a little uncommon, and that might give you some uncommon insights. Do you think that that's true at all?
It's true in multiple ways, and thanks for asking the question. One of them is that it's true that behavior has traditionally been looked at from two completely different angles. The one from psychology, that is interested in behavior, in try to gain insight into human behavior, animal behavior, to gain insight into animal behavior. And the other one from ethology, which is just to try to understand the rules of animal behavior. But I come from a standpoint of a biologist using biological tools that enable to gain insight into mechanism, cellular mechanism, molecular mechanism, try to find causality in behavior in ways that is never achieved or even sought after, neither by ethologists, neither from psychologists. The other point you're making is that basically I come to the field of behavior as an outsider, which
right.
I think is super important. And in fact, it even is a guiding principle of my own lab, which is people who come from my lab all come as outsider of the field of social behavior. So there are people who have worked on copper metabolism or in immunology or in C8 against motor behavior. People from my lab all come from very different horizons, and they come as outsider, and because of this they ask tough questions.
But you're choosing that specifically. I want people like that. Yeah,
exactly.
Yeah. And do you think that's why your lab is so productive?
I don't know if it's the reason my lab is productive. I think I have very good people, but at least that's the type of environment I want to have. I want an environment where people are challenging ideas, including my own ideas.
Yeah.
And I think that's enabled us to make a lot of progress.
Yeah. Okay. I don't know that this is true, but it seems like growing up you had two parents who were both working, both had active careers, and both did parenting to you.
Yeah.
Do you see any parallels between that and the work that you're doing in mice and parenting?
You know, it's an interesting point. The influence of my dad and my mom having the same job, basically, for me, I phrase it in a slightly different way, but what you say might be correct. I think that, my mom and my dad had the same job. They both were parenting. My dad was the person doing the groceries and doing the cooking at home. And so, there was really sharing of both intellectual, professional, as well as personal pursuits. For me, I think was very influential in feeling that I could do whatever I wanted. There was no real impairment in me desiring to do anything I wanted in the same way my brother would. And the reason I think this was very influential is that a lot of my classmates had a mom who stayed at home, and there was no equilibrium, no balance between the achievement of the father and the achievement of the mom. And I remember several times when I went to visit my classmates when I was a child, mom would say, I think it would be great if we could buy this or that. And the child would say, but mom, you don't have a salary, so, it's not to you to decide. And I thought, this is not right? Yeah. And so I think I never experienced that because my two parents had the same salary, the same job, and the same duties at home. And so I felt as entitled to do whatever I wanted with my life as anyone else. In fact, it didn't even come to my mind that there would be a difference between a boy and me in terms of ambition and achievement. In fact, I was always the best in class. So, why would the boy who was not as good as I am would do better than me later? There's just no way. And it's only when actually I became a PI that I realized that, things were not that fair. Right. I never suffer from it myself, personally, but I could see, that going to conferences, for example, the expectation of women giving good talks was not the same as a man. In fact, it has happened to me many times that I would go to a conference and, was the first day and nobody knew each other, and I would sit close to a famous guy, and the guy had no interest in talking to me, and would talk to whoever other guys were at the table. Oh, and it just turns out that I was the one giving the keynote lecture, and the next day that man would come to me and looking a little sheepish and say, "Oh, that was very interesting." Well, yeah.
I mean, did he acknowledge that he was sitting next to you at lunch the day before?
Yeah, totally. And would come to me kind of apologizing because he absolutely did not pay attention, didn't expect that I would have anything interesting to say.
Yeah.
Oh, and I was the one giving the keynote lecture. Oh, and he thought my keynote lecture was really good.
On top of that. Yeah.
And so I think it's, for me, being here at Harvard and being a teacher, an instructor, an educator, I think this is something I always keep in mind, which is that it's fascinating that I have a lot of undergrads in the lab, and I have men and I have women, and they are all super excited by science, and I feel it's my duty to make sure that everyone feel equal and equally confident on what they want to do. It's super important that men, women, people of color, whoever their ethnic background or they come from underprivileged background or whatever, feel that the world belongs to you.
Yeah, if you're in my
them,
lab,
right?
we're going to do science
Exactly.
in here, you're all
Absolutely.
do
I
it.
want everyone excited. And, I think that what is hard is that, is these 1,000 cuts, right? So if you're a young woman or maybe if you're African American or come from an underprivileged background and don't have all the cues of what's around you, you're being penalized.
Death
you're
by 1,000 cuts, you're saying.
Exactly.
Yeah.
And so that's what I worry about.
Yeah.
And, I remember something that I didn't realize. I discovered when I read the book from Sonia Sotomayor on her biography, on her coming from an extremely underprivileged background from Puerto Rico, where her parents didn't even have a bank account, and that she was this spectacular student, and then all the doors opened to her, and she was accepted at Harvard, she was accepted at Yale, and all these places, and she went to Yale, and then she had no clue on how the academic environment works, and she had some guidance, but it made me realize that
you knew that anyway.
Right? I had that privilege, basically.
Yeah. So one final question. I actually don't know the answer to this, but so you've got all this work. You won the Breakthrough Prize, and your work has sort of been tied to both the male and female brain, but also parenting, right?
Yeah.
And I don't know whether you have any kids or not, but if you do, has that influenced the way you think about work? And if you don't, has it influenced the way you think about parenting?
I don't have children. I have a niece and 2 nephews.
Yeah.
No, I don't think it influenced me. And the fact is, parenting was recognized by the Breakthrough Prize, but my interest is way more general about social behavior and instinctive behavior. Similarly, I'm similarly interested in aggressive behavior.
Yeah.
I'm similarly, we've made some really interesting finding recently on the brain response to social isolation, which I think is super, super interesting. Why do we have an urge to be together? So I think more broadly about instinctive social behavior. For me, this is what's driving me.
Yes, you do, but then when the prize comes out, people think, oh, she's working on parenting.
That's true. Yeah. But then now they have a chance to
hear the rest of it.
More.
Yeah. Okay, all right. That's it. Thank you. I appreciate it.
Yeah, you're welcome.
We had been trying to get our schedules aligned for a while, but I'm glad we finally got it done. Super interesting person, great science. Thank you, Catherine, for making time and letting me set up in your office. Okay, this podcast will be archived on thetransmitter.org, where we have also supplied a transcript which includes some links for more information. You can find this podcast on YouTube, Spotify, Apple, and wherever you get your podcasts, and you can subscribe there, and you can rate this show. Some of the information for the intro was taken from Trading Economics, which supplies historical economic indicators for 196 countries, including France. And also in the intro, I quoted from an article titled Budget Woes Cast Chill on French Research, published in 1996 in Nature. If you'd like to comment on this show or whatever we do with the Transmitter, you can find us on the social media platforms X, Blue Sky, Mastodon, and LinkedIn. Our theme song was written and performed by Chris Collinwood. Thank you for listening to Synaptic. Until next time. You know what? If I stack some books up.
Yeah, well, you have plenty of books here. We can pile up. You want to make a pile like this?
Let's try that.